Hello students, a warm welcome to all of you in our 7 days online certification course on topic next generation sequencing. Okay, in today's session we discuss about the sequence alignment. Okay, in today's session we discuss about the sequence alignment. Okay, what is the requirement of okay? So in today's session we discuss about the sequence alignment. So we start today's session and understand what is alignment, what is the types of alignment, and many other prospect of alignment. What is alignment? Alignment means sequence alignment is a procedure of arranging the two and more sequences. Okay, whenever we arrange the sequence to identify the region of similarity. Okay, this similarity may be arises due to the common ancestry, due to the functional and structural constraints. What is alignment? Alignment is a process by which we arrange the sequences. Why we arrange the sequences? Because we want to find out the similar and identical regions. Okay, this similar and identical region is generated due to the common ancestry, functional and structured domain. Okay, if the sequence is match or have a similarity, then it will show line like this. If no if no match means gap, then it will show like this. This is the gap. This is the match, and this is the mismatch. Okay. When the sequence is aligned, then gap may be introduced. This gap introduces the insertion of sequences or deletion that have acquired over the evolutionary time. Okay. In simple language, what is alignment? Alignment is a method by which we arrange the sequence. Basically, in simple language, it is the procedure to arrange your sequences. And this sequences may be your nucleotide sequence, may be your protein sequences. And what is the purpose behind this alignment? The purpose behind this alignment is to identify the similar and identical region. Okay. This similar and identical regions arise due to the common feature. Due to the common feature. Okay. If your sequence have a similarity, then it will contain the similar and identical sequence. If your sequence does not have a similarity, it means it does not have any types of relationship. Okay. In your sequences, if the gap is arise due to the mutation. Okay. And this mutation may be your insertion, may be your deletion. Okay, this mutation, due to these mutations, the species are differ to each other. Okay, let's suppose it is a species A and this is species B. Both the species have a common ancestor, but both have a different characteristic. Why they are the different? Because their gene is evaluated and the reason behind this evolution is a mutation. It may be insertion, it may be deletion. Okay. Now we discuss about the types of alignment. If we're talking about the types of alignment, most of the times alignment is two types. First is your pairwise sequence alignment. Second is your multiple sequence alignment. Pairwise sequence alignment means whenever we align two sequences. Whenever we want to find out the similarity and identity between the two sequences, then this is known as a pairwise sequence alignment. Whenever we want to find out the similarity more than two sequences, then this is known as a multiple sequence alignment. Okay. This pairwise sequence alignment are divided into two parts. First is your local alignment and second is your global alignment. Local alignments means whenever you want to find out the similarity into a specific region. Specific region. And in global alignment, whenever you want to find out the similarity entire sequences. Okay. For global alignment, the algorithm behind the global alignment is your Needleman Wunsch algorithm, and the algorithm behind the local alignment is your Smith-Waterman algorithm. Okay. Now we discuss what is the importance of alignment in NGS. This is the common to all, both NGS or normal nucleotides or protein sequences. But now we understand why this alignment is so much important in case of NGS. Because after the sequencing, NGS machine provide a million and billions of short reads. Okay, millions and billions of short reads, and the size is approximately 50 to 300 base pair. Because before the NGS, we fragment out the large genome into a small fragment. Okay, and this process is known as a library preparation. Sorry. Okay, these reads are random fragmented of the sample's genome. Okay, then to find out the exact order or to find out the match and mismatch and deletion, we align these results with reference genome. Okay. Here first we take the reference genome and our NGS data, then we align. Why we align? Because we want to find out the exact location of your gene. And what is the benefit behind this? The benefit behind this, we easily find out the gene, easily identify the gene, we easily find out the mutation. We easily find out the conserved region. We easily find out the mismatch, insertion and deletion. Okay. Alignment in NGS. After the NGS, millions and millions of short reads are the NGS machine produces millions and billions of short sequences. Then we take this sequencing and align the sequences with reference sequences. Why? Because we want to find out the exact location of your target sequences. Okay. Now, why the alignment is so much important? Because we want to identify the genetic variations like as a single nucleotide polymorphism, insertion and deletion. We also want to measure the gene expression. We want to find out the mutations, pathogens and novel sequence. Perform the genome assembly and comparative genomics. This is the purpose behind the alignment in case of NGS. Okay. Now, types of alignment in NGS. In NGS, alignment is two types. First is your reference based alignment. Second is your de novo alignment. Reference based alignment means you have a reference sequences, known reference sequences. And the known reference sequences like you have a reference genome of human, mouse, and any model organisms like C. elegans, zebrafish, etc. Okay. If you want to perform this reference based aligning, then you use this software like BWA, OT2, and HISAT2. How we use this HISAT2, we discuss this our incoming slides. Okay, and star. These are the tool to perform the reference based alignment. Okay. Next is your de novo alignment. De novo alignment means whenever you does not have any reference genome. If this reference genome is not present, then you use this de novo alignment. Okay, read and compare to each and other to reconstruct the genome. Okay, and we use this tool like SPDS, Velvet, and SOAP de novo. These are the tools that are used to perform the de novo alignment, and these are the tools that are used to perform the reference alignment. Okay, these are the two types of alignment in NGS. Okay, now we discuss how the alignment will work. Okay, first we compare the read with compare a reference genome. Okay, and find out the best match. Okay, chromosome, and we also record the chromosome position and their match and mismatch and gap region and mapping store. Okay, let's suppose this is our read and this is our reference sequences. Now your read is A, P, C, G, T, A, G, C, G, G, A. Now your reference sequence A, T, C, G, T, A, C. Then A, G, A. Here A. This is match, 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 and this is the mismatch. Again match, again match. 1, 2, 3, 1, 2, 3, 4, 5, 6, 7, 8. And this 8th position is a mismatch. Okay. In this way, we easily find out the match, mismatch, and gap while comparing your read with genome with reference genome. Okay. Now the alignment tool and common command. Like if you use this BWA, then it is used for the DNA sequencing reading map, and this is the command line. We use this command line on Ubuntu. Okay. For OTK2, we use this fast and OT2 we use fast alignment for short read, and this is the common command. If we use HISAT2, then it is the RNA sequence aligner, and this is the command line. We use this command line for aligning the sequences. Next is the star. It is a high speed aligner for the RNA sequences, and this is the command line. Okay. Now we discuss the output alignment. Output alignment, first we submit our FASTQ file and reference genome. Then it will convert the SAM file. SAM file means human readable. Then we convert this into the BAM file. BAM file means binary composed version. Okay. It will contain the read name, mapping position, quality score, alignment score, and CI registry. Okay. Then after completing the alignment, we will go to the sorting and indexing with BAM file, variant calling, and visualization in IGP. Okay. To convert this BAM file to SAM file, we use a software that is a SAM tool. Okay. This is the theory portion behind the alignment. Now we discuss this is the theory portion. Now we discuss the demo part of alignment. Now we discuss the demo. Okay. For this, first we simply search Ubuntu. Okay. This is the home page of Ubuntu. Now we discuss how we use this command. So first we open the conda environment. So simply click conda, activate space RNA sequence. Rewrite conda activate RNA, enter. Okay. Now environment is created. Now we move to the documental. So cd space /mnt/d/ enter. Now you are in D drive. Now ls in your D drive. These are the folder. Our required folder is a DNA barcode. Then cd DNA tab R. Now we are in this folder. Now ls in this folder. These are the file. Our required folder is the FASTQ NGS. So simply click on cd space FASTQ space and gs and enter. Now these are the file are present. Okay. Now we run the HISAT2. For this, first we write HISAT2, then space -x, then hg38 space then genome space -u, then your sample name. This is my sample, and this is a gap between the sample space, only one space, and then again space -s, then sample dot sam, and enter. Okay, and wait for the result. Whenever this mark is blinking, it means this software is running. Okay. Now we understand how we perform the HISAT2 software, and the HISAT2 software is used for the alignment process. Okay. For this, first we create the first we open the so first step to create a conda environment and to so now first step we create a conda environment. For creating the conda, now first step is to create a conda environment. To create a conda environment, we simply search conda activate RNA and enter. Okay, now this environment is created. Now we move to our design. Now we move to the DNA barcode folder. So simply cd space mnt dash, this folder is in G drive, then ls. Okay. cd space DNA tab, enter, ls. Then we want to move in FASTQ NGS folder. So cd space FASTQ NGS. Now we are in this folder, ls, and these are our file. For running the HISAT2, first we write HISAT2 space, then HISAT2 space -x, then space hg38. These are the reference sequences. Then genome space minus capital U, then your sample file. My sample file is this one. Okay, then space minus, then space minus S dash S space sample dot sam, and wait for the result. Whenever this alignment is completed, then to complete this, okay, and this process will take time approximately 5 to 10 minutes. Okay, the alignment task is completed. Whenever the alignment task is completed, then this will show. Okay, your reads, your unpaired 100% completed, that's it. Okay, and whenever we see now, check this file in the in FASTQ CNC format and in energy format. Now we check this folder. So checking this folder, simply click on ls. Now in our folder, this in this folder, that is the FASTQ NGS, this file is present, that is this file. Okay, this is our alignment result. Okay, now for the further analysis of the sequences, we convert the SEM file into the WAM file, then short this WAM file, then convert it into the indexing. Then we use this file for the visualization as well as for the feature count. Okay, now we perform this task. For this, first I click on Control L. For the conversion, we require a software, that is the SEM tool. Okay, and how we install the SEM tool? By using a command. And what is that particular command? I will discuss. By using this command, by using this command, that is the sudo apt get install SEM tool. Why? We easily install this software in our PC. Okay, then okay, now whenever this software is installed in your PC, then you easily convert the SEM file into the WAM file, then short your WAM file, then indexing. For SEM to WAM, we use this command, that is the SEM tools view minus vs aligned dot SEM aligned by WAM. Okay, I will show you how to do this. So simply click on SEM tools space view minus v minus vs, then your sample name, that is this one. Copy this, paste, then this one, then sample dot bam. Okay, then enter and wait for the result. Okay, this is the command, that is the SEM tools minus vs, then SEM file in which you want to convert, we convert this into the WAM file. Okay, and wait. Whenever this cursor is blinking, it means your task is on running mode. Now our SEM file is converted into the WAM file. To checking this, you simply click on ls. And now this is your file. This is your SEM file. Now this is your WAM file. Now we short this file. Now for converting, for shorting this command, we use this line. Okay, SEM tool shorted SEM dot sample dot bam minus out office output in sample dot bam. Okay, now next task is to short this file. For shorting this file, we use this command, that is the SEM tools shorted sample dot bam space minus o space sample dot bam. Okay, so write down SEM SAMtools short, SAMtools short, then sample.wam, this is the file, and space -o space sample.wam and enter and wait for the result. Now merging from two files and one file is memory blocks. Okay. Now your file is shorted. Now checking this, you simply click on ls. Now this is your okay, now sorting is completed. Okay, now we convert this into the indexing. For the indexing, we use this command, that is samtool index sample dot short. So write down samtools space index dot sample dot short and I think there are some issues again. Copy and paste and enter. Now we again we convert the WAM file into the short file. So I using this command, that is the samtool short sample.wam minus one output file is sample shorted.bam. Here is a mistake is this one. Here we give the same name, that is the sample.bam and sample.bam. Okay, but we give the another name, that is the sample. Now task is completed. What is our common mistake? Common mistake is this: we give the same name, output or input we give the same, but we give the different. Output is your sample sample file dot bam, and our output input is our sample dot bam, and our output file is a sample dot shorted dot bam. Okay. Now this shorted file is installed in our, now this shorted file is created. To checking this, we simply click on ls. Whenever we click on ls, then this file is shown. This is the file. First is your sample file. Then we convert the sample file into the BAM file. This is, then we sorted this BAM file. For sorting this BAM file, this is the command. Now we perform the indexing. For the indexing, we use this command, that is the sample tool dot indexing. Copy, paste, and my name is shorted dot bam and enter. Okay, then again check ls and enter. Now this is our bam file, this is our sam file, this is our bam file, this is the shorted file, and this is the indexing file. Okay, now the alignment is completed. Okay, now this file is prepared for the further analysis, means for visualization for the visualization and for the feature count. Okay, in next lecture we discuss about how we use the feature count software. Okay, now I will discuss one and most important point. Here we use the reference sequences. Here we use the reference sequences of humans. So now we understood how we download that particular reference sequencing. Okay, for downloading this particular reference sequences, there are different databases are available, but I am use the database of HISAT2. Okay, so now open this. Now we during the alignment we use a reference sequences. Now we understand how we download these reference sequences. For downloading these different sequences, there are a number of softwares are available, number of databases are available, like your NCBI, UCGC browser, ENSMBL, and HISAT tool, and your Bold database. But in today's session, we discuss about how we download the reference sequences from the official website of HISAT2. So for this, in Google, uh, we simply search HISAT2. This is the URL, and I open this. This is the homepage of this URL. These are the different. This is the homepage of downloading the different reference sequencing. Okay, this is for the humans, mouse, C. elegans, and Saccharomyces. 8 बज रहे हैं शांति से थोड़ी देर और सो जाओ. Okay, these are the different reference sequences: Homo sapiens, mouse, C. elegans, or your Saccharomyces cerevisiae, and these are the binaries. These are the indexing humans, genome. This is for downloading this. This is the URL for genome. SNP. This is the URL. This is the URL from the UCSC browser. Okay, this is the requirement for the humans, musculars, C. elegans, and other binary sequences. Okay, by the help of this page, you easily download all these reference sequences. Okay, and what is the link? I'll WhatsApp this link on your WhatsApp group. Okay, now whenever you click on this main, then you give the then you know the complete information about this HISAT2 software. Okay, if you want to download the manual of this HISAT2 software, then simply in Google you simply search HISAT2, then open this home page. This is the HISAT2 now home page. Here the complete information related to HISAT2 is available. Whenever you click on this manual, then you get the complete information about this HISAT2. What is the HISAT2? What we obtain from the HISAT2? What is the running command of HISAT2? And what is the mean of each and every now, and we also know. And this is the complete command line. Now what is the mean by X? X means this is the baseline of indexing reference sequences. Minus, if you use more than 2 sequences, whenever you align more than 2 sequences, then we use this -1 and -2. Minus U is for the command separated line. Okay, and here the complete information are available related to the HISAT2 software. Okay, in this way you easily perform the alignment process. For the alignment process, you require the reference sequences or your sample sequences in a FASTQ format. Then and you download the HISAT2 software as well as SAM2 software. HISAT2 software is for the alignment, and the SAM2 software to converting the SAM file to the BEM file, then BEM file to shorted the file, then indexing. Why this is important? Because this is important to further analysis. Let's suppose we want to understand the feature count and visualization, then we use this, shorted file. Okay, and this is all about the today's session. If you have any doubt, please write down on comment section. Thank you so much.